I think we can get started. I realize that a couple of faculty are out either for the ASH meeting, the lymphoma meeting sort of, and also some of them are out because of the San Antonio breast symposium. So today we are very fortunate to have Dr. Chae, who is a professor of medicine in the Division of Hematology Oncology and co-director of the Early Phase Clinical Trials Unit, as well as the co-director of the Developmental Therapeutics Fellowship Program. He obtained his MD from the Seoul National University College of Medicine, and then came to the U.S. and earned both an MBA and an MPH at the Johns Hopkins University. He went to Albert Einstein for his internal medicine training, and then a fellowship at MD Anderson in hematology and medical oncology. In 2014, he joined the division, focusing on early phase clinical trials work and research. As many of you know, his interest is in thoracic oncology and precision oncology. He has led many targeted therapy clinical trials and immunotherapy combination trials, applying both tissue and blood-based biomarkers for some of this work. He has been the PI for the NCI Match trial, and as well as and takes leadership role in the SWOG DART trial. And related to the DART trial, the title of his talk is The Lessons We Have Learned for the Immunotherapy Basket Trial for Rare Cancers, the DART. All yours, Dr. Chae.
Thank you. Thank you for the opportunity to talk about this topic that's dear to my heart. So we're going to talk about the lessons we learned for the DART trial. So the DART stands for dual anti-PD-1 CTLA-4 therapy for rare tumors. And I've been working as a vice chair of the Early Therapeutics and Rare Care Committee at SWOG, working also with Dr. Patel Kurjak, who are co-study chairs for this trial. And I've been also appointed as a translational medicine chair. So I've been working with CMAC, NCI, CTAP, on a lot of translational projects that's coming out from this study. So we're going to talk about all of that during our talk. This is my disclosure. So why did we decide to tackle rare tumors? So as a group, they're not really rare anymore. A quarter of all cancers diagnosed in the U.S. are actually in the bucket of rare tumors. By definition, we usually use less than 6 out of 100,000 per year incidence. And usually we have targeted therapy that are targeting the genomic or other predictors of response. But in rare tumors, the whole disease is rare, and if you make it molecularly rare, it's not really feasible to target them anymore, unless you do a pan-tumor study. So here, at this time, when I went to SWOG, so I imagine myself just being a first-year faculty member, and I want to I have a question, where do I build my career? So at that time, Northwestern has become SWOG member. So, we've been traditionally ECOG, but then let's go to SWOG and see what I can do there. And I went to their early therapeutic committee because I was the first faculty to be hired for the Developmental Therapeutic Institute at that time. So there were a lot of discussion about how do we bring the success story of immunotherapy. At that time, melanoma, we had success story of the nivo-ipi working regardless of the PD-L1 at that time. And there are many more success stories following rare cancers like Merkel cell, Hodgkin's lymphoma, so forth. So maybe the immune system doesn't discriminate histology, and maybe we should make a basket trial that will really explore how the combinatorial immunotherapy works in those tumors. So I met with Dr. Kurjak, who was the SWOG chair at that time, and Dr. Sandeep Patel at UCSD at that time had the same idea. So Dr. Kurjak said, why don't we, 3 of us, team up and make a trial? And it took almost 3 years to really set this trial and up and running. So this is how we came up with the idea. So 3 of us, and with the Megan, who's the statistician, worked hard on developing the concept. So we made it very simple. So it's the nivo-ipi 1 mg per kg, not the 3 mg that's used for the melanoma. So it's much less toxic and it's given every 6 weeks. The Nivo is fixed dose every 2 weeks. So this is a regimen used for Checkmate 227 in lung cancer trial, and their toxicity rate is much, much less than the one we've seen in the melanoma trials. So we use this regimen, and we use the Simon 2-stage design, so 6 plus 10. And if we see more than 2 responses, then we would go to the next expansion cohort. And at that time, we were told no. The resource allocation. So we were told to wait. And at that time, NCI match trial was ongoing. I was the PI of the arm W, which is the FGFR. It was the AZ drug AZD4547 targeting FGFR124. It was a good trial. We published the results in JCO. We learned a lot, but then because this trial was accruing about 25% of patients that are rare, they thought this was competing with DART, and they said, no, let's accrue to NCI match first. But then they ran out of the money to do NGS, and they opened their genomic profiling to other commercial payers, the platforms. And at that time, they also needed a space for people who failed or failed screening or progressed on NCI match. So we had this strategic marriage of these two trials. As you can see, if you screen fail or you progress on NCI match, and they belong to this category of rare cancers, as defined as in our protocol, they would be eligible for our trial, DART S1609. So we had this strategic partnership or marriage, if you will, and we were given green light to open this trial. So first, it was approved by SWOG May 2015, and then we activated a trial in January 2017. So initially, we had 33 cohort, and we had investigators from all different trial groups. Like ECOG, we had Dr. Sylvia Lee, who was a PI of the metaplastic breast cohort. So this was an ongoing collaboration within the NCTN, and we had lots of amendments of the protocol incorporating like new cohorts. February 2018, we were able to get funding from CMAC to add biomarker studies, and we were awarded the pilot grant that allowed us to secure tissue as well as all the blood. And our first cohort was, the result was presented in April 2019, neuroendocrine cohort. It got into the New York Times, and it was a big thing at that time. So high-grade neuroendocrine, if you look at them, 44% response rate, 8 out of 18. We didn't see any response in low-grade neuroendocrine. This was published in CCR. And then we presented our salivary, small bowel, and so forth, angiocarcinoma, thyroid cancer, and we closed to new enrollment in 2023. There are still patients on this trial, ongoing response, and you wouldn't believe it. So the final patient accrual was 798. It was open across 1,015 sites across United States. So people had doubts, can you accrue to rare cancers? We over-accrued, we did really well. And a lot of sites where the rare cancer patients hear about the hypes of immunotherapy, they never have really access to immunotherapy. And this was open in all NCTN, pre-COG, all community sites. So we had more than 50% community sites, physicians accruing to DART. And these were the initial 33 cohorts, and as you can see, these are rare tumors by their incidence. And what's interesting is we have neuroendocrine carcinoma cohort that is regardless of any tissue origin, but then we have the carcinoid of lung as a separate cohort. We have unknown primary as one cohort. We have not otherwise categorized NOC category. And here, we've accrued the tumor types I've never heard of. And and they became a separate cohorts later on. If they reach more than 5 patients, that we would make a separate cohort. We would amend protocol. So, and then if there were investigators really interested in looking at subtype of each cancer, like breast cancer is common, but not the metaplastic carcinoma. So you would, we had Dr. Sylvia Lee suggesting this cohort, so we would add it. We had the PICOMA, atheneoblastoma. It's a olfactory nerve bulb tumor. I didn't know about this tumor, but this just became, they coming to our trial, and we were accruing, so we made a separate cohort. So there's a lot of different types. And then we had another cohort that we generated. It's a small cell carcinoma of ovary hypercalcemic type. Apparently it hits women in 20s, 30s. And there was a New York Times article that, with nivo monotherapy alone, there was deep durable response. So we added this patient, and we actually published this data. We actually saw really good response there. And then we made one cohort that's biomarker based, so PD-L1 amplified cohort. So this was by NGS, by the copy number, 7 or above. And then we added angiosarcoma. We wanted to add rare sarcomas, but there was a separate sarcoma trial ongoing with NCI. So it was competing, so we couldn't accrue anybody, but angiosarcoma was allowed. And then we added small cell transformed prostate neuroendocrine tumor later on. So this is, you can still go on to SWOG, swagstat.org. It will show you how many patients are accrued to which cohort at what time point, and which cohorts are still open and which cohorts are permanently closed. So these are the sites that opened our DART study across the United States. And we actually saved lots of patients. This is a Crane's Chicago business. The reporter came to me and asked, how did it all come to be? And we had one adrenal cortical cancer patients in early 30s, progressed through all conventional therapies, and later on really had deep durable response, still ongoing CR. And we never thought, adrenal cortical would work. So definitely there's some group of patients that do respond to immunotherapy. We just have to know who they are, so that we are doing the immunotherapy, the translational medicine pieces. And then this is one of our own developmental therapeutic patients who's an esophageal neuroendocrine tumor, deep durable response. And she did really well. She passed away from non-oncologic reasons, but we were cheering her up with all these awards and ceremonies, and she did really well. So here are the schema of the trial cohorts and the total patients and the objective response rate, and there are as high as 29% and some in 17%. So as you can see, these are the tumor types that are traditionally considered cold, immune desert. And indeed, if you look at them, they are immune desert. A lot of them are still immune desert. But somehow, I believe CTLA-4 is bringing immune cells from the lymph nodes nearby. So most responses were lasting more than 2 years, surprisingly. And many are still ongoing responses up to this day. So this is first neuroendocrine data. We've shown that only the high grade, as you can see in the red waterfall plot, that they had deep durable responses. And then because of it, we had to make a separate cohort for just high grade, and which was confirmed to be the same responders. So this was later added to the NCCN guideline treatment options. So angiosarcoma, mostly UV related, more in the facial, or scalp, or the ones that probably had high TMB and did respond very well with deep durable. And this is a representative figure of the facial lesion that disappeared with the nivo-ipi. And it did get into the NCCN angiosarcoma treatment guideline. And for the rare tumors, look at the vulvar cancer. There are a group of patients who did bond with some durability. Small cell ovarian cancer, hypercalcemic type. We did find 2 responders, 1 still ongoing. Clear cell ovarian cancer is a rare non-epithelial ovarian cancer that showed a lot of good responses. So we are very interested in really deep, delving into who they are. And we're still doing the translational piece. Granulosa cell tumor, which is another non-epithelial ovarian cancer. We think they're hormone-driven, but we've seen that some of them really responded very well with much less toxicity. This is a metaplastic breast cancer, and these 3 patients have had still ongoing, very deep, durable responses. So we're looking into this, and I think based on this, this will soon get into the NCCN. Bronchoalveolar cancer, as you can see, there are a few that did respond with durability. Thyroid cancer, what's interesting, we did have some idea that anaplastic can have some immune infiltration, and they did respond very well. But some of the patients are cold tumors, immune deserts, well-differentiated thyroid cancers that still responded well to nivo/ipi. So we're very interested in this. So this paper, we submitted to JCO. It will come out soon. Salivary gland cancer is also a rare cancer, high unmet need. You do see a subgroup of patients who have deep, durable response, but mostly non-adenoid. Adenoid, as you can guess, most of them didn't respond, but one. One patient had a deep, durable response, and we don't know why. In basal cell carcinoma, there are some patients who did show good response, as you can see here. PNET, pancreatic neuroendocrine tumor, not as great as non-pancreatic, but we did see some responses there. This was all published in CCR. Vaginal cancer, you do see some responders there. Gallbladder cancer, as you can see, some of them have good responses. And these are MSI, stable cancers. And we added gestational trophoblastic neoplasia, and it was really hard to accrue them. But 4 out of 4, 3 had really good biochemical response, clinical response. They don't use RECIST. So we published this in CCR as well. Adrenocortical, as I told you, there are subgroup of patients, and these seem to be still immune deserts, but they did respond. And desmoid tumors, same thing. We think they're immune desert, but some did respond and ongoing responses. So really, the next question is why, and what are those biomarkers, or what are their traits? And we did find unique cases where they had dual PDL1, PDL2 amplification with high PDL, high TMB burden, and this patient responded really well. So we have, lots of publication coming out from different angles. So as a group, we also found that first cycle toxicity correlated really well with survival in this rare cancer population. And here you can see there's the black curve is the overall survival of a patient with no side effect. And of course, if you have severe side effect, it affects their survival. So that's the red. But if you have just grade 1 and 2, they did really well in terms of survival. But if you take out the dermatologic side effects, with no skin side effects is the one on the right side, darker blue, and the lighter blue is the one without the one with just the skin side effects. So there's something about this mild toxicity, especially the dermatologic ones, that is associated with better prognosis. So here is the next question we've been asking. What can we learn from DART? And from the translational medicine side, what can we do? So we all remember all these TCGA papers coming out from breast, lung, and we're learning the genomic landscape, transcriptomic landscape, and we really don't have that kind of landscape papers from rare tumors. So we now got the phase 2 CMAC NCI pilot grant to look at really the rare cancers that we were able to have tissue for and really learn the genomic and immune landscape. And then also, what are the response predictor? And what are the predictors of immune toxicities? So we can look at all that within the DART platform. So first, we have the first 1/3 of patients who are already enrolled in NCI Match, so we can utilize the existing NCI Match platform database. And then for the DART, we have tissue and then blood. Blood we stored at baseline, at first imaging, and at progression. So we have 3 time points blood, and we've collected the common biomarkers, but we also got the germline, we got the proteomic immune signature from the blood, we got ctDNA, and we have the CMEC and the CIDC support system that is part of the CMEC pilot grant. So because of the lack of the tissue, we have tier 1, which is always the priority, and then tier 2 and tier 3. So we try to do all whole exome and RNA-seq. If RNA is not enough, just nanostring, CyTOF, multiplex IF, and from the blood we are using the Olink platform provided by Mount Sinai. So these are the collaborators. It's a true team science. We're still doing the telecom meeting every few weeks to get this going. And we've been publishing and also presenting these biomarker works. So the first one comes from the neuroendocrine cohort, with cohort 23 is the all neuroendocrine, and cohort 52 is the high-grade only signed cohort. So from that, we have nanostring, whole exome sequencing, PD-L1, and some special immune profiling using multiplex IF. And this is in collaboration of Kara Haymaker from MD Anderson. So here we also looked at the cytokine, chemokines on soluble proteins using Olink. And then we also did the CyTOF looking at the change of immune cells, and also did the TCR repertoire analysis. So PD-L1, you can see, did not really correlate with any of the responses seen or the survival. And from the nanostring, we were able to see that B-cell and dendritic cell signature in the tumor tissue was showing somewhat correlation with response and survival. And this has been consistent with the recent literature supporting the importance of lymphoid aggregates, or TLS, in the tumor. So here is the TLS signature found in the tumor, and it did nicely correlate with response among these all neuroendocrine cohorts. We looked at the immune infiltration, and it did not really show good correlation by clusters. And responses are seen both TIL excluded and TIL included in high-grade tumors. And through Olink, we did see that this combination therapy promoted some immune mobilization. And then there are some discrete EM T cell subsets that differentiate response at baseline and early on treatment using the CyTOF. We looked at the thyroid cancer cohort as well. Again, the PD-L1 nor the immune cells correlated with response. But we don't know what this means, but double positive PD-1 and PD-L1, double positive TILs were correlating with response in this cohort, but still very small numbers here. And TMB did not correlate with response in this cohort. So really, I mean, we have the metaplastic cohort, angiosarcoma. We have everything lined up to sort of answer questions. And once we're done, we're going to do a pan-tumor analysis looking for different biomarkers. But really, the goal is to building the rare cancer TCGA out of the DART trial. So what we are also doing is we're looking at the mass spec-based serum proteomics. We're looking at the AI-based radiomics from the CT DICOM file we gathered. We have the H&E slides as well as the multiplex IF. So we're using the digital pathology AI pathomics to analyze those data set as well. We'll do the multi-omics analysis, creating a model for that. So this is data from my own lung cancer clinic where we use the mass spec-based proteomics to see if that they correlate with response or delayed time to toxicity. Because remember, these proteins are mostly coming from the host patient liver, not from the cancer cells. So they do affect the host immune milieu. And this is the radiomics work that we are doing with the DART patients as well. And it doesn't have to be AI-based. And this is a very simple analysis that Megan, our statistician, did with our patient. This is just looking at the change in tumor size alone at the first scan correlated really well with 1 year overall survival. It's almost linear. And you can see that RECIST is 30%, 20%. There's cutoff and it's categorical. But if you just break it down by the 10% interval of the tumor shrinkage at initial tumor assessment by the scan, it does correlate really well with the overall survival, at least in our DART cohort. So we thought that was very interesting. So what comes after the DART trial? So we are working on the DART-2. So histology agnostic, but has to be rare tumor, biomarker agnostic, so PD-1 and LAG-3 combination approach is being pursued currently for DART-2. And then we are also thinking about a biomarker-based trial. So the Nivo-Ipi is ran out of patent. The BMS is not interested in developing that combination anymore. But there are many other agents that are targeting it, whether it's bispecific, whether it's combinatorial. So we designed a tumor-infiltrating lymphocyte-directed anti-PD-1 and CTLA-4 therapy for rare tumors, but unexposed to immunotherapy. So the acronym is TASTE. Let's give tumor the taste of immunotherapy. And this is based on our paper published in JITC. And we found that even the tumors that we thought were traditionally immune desert had about 20% of immune inflamed cells. And these were not just by nanostring or immunohistochemistry, but they are just manually, pathologists manually just finding lymphocytes within the tumor microenvironment and doing the whole slide-based, AI-based annotation of TILs. And then across different tumor types, if they got immunotherapy, if they have higher percentage of TILs, as detected by the AI TIL assessment, there's nice correlation seen in survival rates. So based on that, we are trying to come up with a biomarker, which is the TIL or the TLS. That could be the biomarker to apply the immunotherapy for those patients. So the future is that we initially started with MSI high. So it's a Pembro. Remember that only required the first Younglee was I think Dr. Benson invited her to Northwestern. I met her when she was here. There was first the philanthropy-funded trial of MSI high tumors, about 100 patients, and that was what eventually led to the approval of MSI high pan-tumor indication. And then came the TMB high. So I think it's about time that we get the TIL high. We just didn't have the right platform to measure TIL, nor the TLS. It required, in order for pathologists to manually just at high power field find those TIL infiltration or the TLS, it's just labor intensive, cannot be done without the help of AI. So I think we are there. There's a lot of companies rising up to do digital pathology. I think the future is that we will get to the TIL high indication for pan tumors very soon. We just need a better agent with less toxicity. So we're working hard on that from the SWOG standpoint. So the take-home message from the SWOG S1609 DART trial and its TM efforts are that it did accrue really well. 798 patients from 1,015 sites, 62% were from community sites. And there's still high unmet need. So we really asked NCI to make this a platform trial, like in Breast I-SPY, but we were turned down. So we're still persistent, but we are pushing to make DART2 trial a reality. But we'll see. And then we're really learning from multi-omics translational medicine effort from the DART tissue and blood samples. And the CMAC, CIDC, the SWOG team science is really in action, and I'm really excited that this will pan out to more discoveries and more precision trials. So stay tuned for making the portraits of rare cancer TCGA from this DART cohorts. And the last but not the least is for personalization of combination immunotherapy, AI-based comprehensive biomarker discovery and modeling can really be applied at this point of the era, and really we can utilize multi-layered data points. I think at every trial we need to be able to collect all these layers of information: proteomics, radiomics, pathomics, and phenomics, of course. And we were, it was a blessing to be able to do that from the DART trial platform perspective. So thank you for listening, and I'm happy to take any questions. Mm.
Kind of off-label, based kind of on that. And we have, these tumors progress like after platinum etoposide, they're just horrendous. And we have young patients that have responded to this and maybe that have been cured. So it's very, very, very important work. The other question was about granulosa tumors. I don't believe it. I don't believe they respond. Because these are the only tumors I've done TMB is not 1 or 2, it's 0.
Yeah.
So how can they respond? I just don't believe it.
Yeah. We don't have good explanation.
They don't respond to anything.
Their PD-L1's low, their TMB is very low, they're hormone driven.
I'm like, this is wrong or something.
Yeah, but it is real, and they are responding, and I've checked the data myself. So yeah, those are real, and we need to understand why. And yeah, I thought the same. Yeah, you're not alone. Thank you. Right. So we think, so when I say immune desert, the TIL concentration was very low. When you do stain for CD8, all of that, it's very low. And some of them were the highest durable responders with the combination therapy. I typically don't think they would respond to PD-L1 monotherapy, but I think because of the CTLA-4 component, that allows you to bring some lymphocytes from outside the tumor. So we've seen that it's not just DART trial. It's the trials that involve CTLA-4. We've been seeing that across different tumor types. But just to see that, but we also saw that it was not the absolute amount of the T cell already infiltrated, but it was more if they had slight structure of lymphoid aggregate or the B cell, T cell, dendritic cell, forming germline cell like germ cell like germ cell germinal center like TLS structure there, then they're the one who seem to respond. So there's something going on. It's not just either are they inflamed or not inflamed. Yeah, and I think that has to do with the CTLA for biology.
So you think the spatial niche or the structure,
Mm-hmm.
like cloud, is the determinant of this response?
I think TLS is important, but not in every histology. Neuroendocrine was important, but in some tumors. But again, it's hard to say because of the small sample size. So I would say in general it's important, but it could be histology dependent. But in tumors like thyroid cancer, where it's well-differentiated thyroid, and really no TIL infiltration that we've seen, but they still did have good durable deep response with the combination therapy.
So the last question,
Yeah, yeah.
promise about last. So for the TCR sequence, you did seems like TCR sequencing there, right?
TCR, yeah, sequencing we did see. Yeah, I didn't bring it up because it's a lot of data, but they've seen some correlation in responders. There were more clonalities, and yeah.
Maybe a question for you. You said 60% of your patients that are enrolled with immunity.
Yes.
How good is the infrastructure for collecting the specimens and doing blood studies?
Yeah. So it's getting samples and getting blood, and the rest is. So we didn't do the real-time central validation of cohort because if we do that, it's such a lag and it's time-intensive. It's a lot of cost, so we didn't do it. So one of my job as a PI was, the community site just, enrolled wrong patients to wrong cohorts. So I ended up, doing a lot of organizing new cohorts and all that. So they're not perfect, and I think that level of proficiency is lacking compared to academic centers. And then they never could get the NGS amplified PD-L1. They would just put patients who are IHC3 positive or 90% on this cohort. So they were not eligible, but we had no mechanism to exclude them real time. They just enroll and they start treatment. That's how the NCTN trials usually work. So we had some problem with that in community sites. But overall, I think there's more plus just getting those patients from the community sites. But yeah, there needs to be more education. So biopsy was just from the archive tissue done at diagnosis. We didn't do any more biopsy because it was so, unless you do like phase 1 trial, it's hard to do any on-treatment biopsy with any community sites. So it was cold before starting any therapy. That's all we can say. But most of them did not get any immunotherapy, just regular chemotherapy. And these are rare cancers, and you'll be surprised for some rare cancers, there's no approved systemic therapy. So sometimes they got this as a first line because, like Desmo, you would get surgery, and just had, no really good chemo options. And they don't want chemo. They know the data's not that good, so they just go straight to the immunotherapy.
Maybe a follow-up question. Yeah, for DART2, you chose PD-1 LAG3. The rationale for that, I'm sure, I think there are lots and lots of checkpoint coming in. Which is the right correct partner for PD-1?
Yeah. So we chose LEG-3 for just practical reasons because it's the only next available one, commercially available. But I think other checkpoint can be equally important, but we just don't know that much. And LEG-3, there's some data that I see LEG-3 is important, but again, like in the case of granulosa cell tumor, we never know unless you actually try it. And these are patients who have no way of accessing immunotherapy, even just the single PD-1 agents, and unless you have a doctor who wants to do off-label, but it's really hard to do that in community. So I think having this type of trial available for community cancer patients who have rare cancer I think it's really important. Yeah.
Any other questions for Dr. Chae? Maybe Dr. Lau? You have a question. You texted me something.
So I'm interested in looking at underlying predisposing conditions, particularly hematologic conditions, and how they impact reactions to immunotherapy. And so seeing the data that you pull together with paired specimens, blood specimens, and tumor specimens, the opportunity to evaluate potentially protocol clonal myelopoiesis or other underlying heme changes, and then tracking immune toxicity seems like this data set would be a great match for that kind of work. And so I messaged saying, I wonder if I could get access to this data somehow. So just wondering, how you guys, if this data is available for other work that might be able to bring it on.
Yes. So this data will all made available for future investigators. So I think it's not just the DART. Any SWOG study or NCTN studies follow the rule that the first paper will come out, and then for TM, first paper comes out, then we'll open for proposals, and we'll and then we'll have limited sources, but we will assess, evaluate, and work with any good ideas that comes. And I do agree that clonal hematopoiesis is a really important biomarker now, not just for hematologic diseases, but also for solid tumors. And ctDNA picks up so much data on that. So I think it's a really good data to explore that question. So yeah, thank you.
Dr. Chae, it's been fun to watch how you've done this over a decade of your life. I think there's a push for pragmatic trials. DART too, but doesn't that fall into pragmatic for rare? That's how you guys, that was really that you have to develop a protocol and open a site, thyroid, specifics. What has been your feedback?
Yeah. So in lung, we are very, accustomed to the pragmatic. That's the title of the trial. It's for the the second line trial that we had open in our cancer center too. But I have mixed feeling. I like it because it's so easy to accrue. It's patient friendly. And the eligibility criteria, exclusion criteria, almost none. There's just principles, and you can get those patients on it. And it's really this clinical progression, and the study investigators don't have even the CT scan results. They don't have DICOM file, no CT. So I was going to work with the Pragmatica trial, but there's no CT data. There's nothing to do any translational work. It's just by the clinical progression. So and then you can never collect any, tissue or blood or anything. It's just go by the book, and which is very simple. So I feel like the DART2 won't be a pragmatic trial, but it will at least collect the CT scans and tissue and blood as we've done. And that's the only way we can learn from rare. I think the rarer the cancer is, we just have to collect so many data to learn. But if it's a common tumor and it's just, we know that the concept is right and we want to validate it, I think we should do pragmatic trials. So, yeah, thank you.
Any more questions for Dr. Chae?
Thank you.